Bismillahirrahmanirrahim. Dear students of 4th year class, today we will discuss the classification of renal cysts. First of all, we will discuss the classification. We will just enumerate and then we will explain one by one. It's multicystic renal dysplasia, polycystic kidney disease, and including the autosomal dominant polycystic kidney disease and autosomal recessive polycystic kidney disease that are included under the heading of polycystic kidney diseases. Next are the medullary cystic diseases, including medullary sponge kidney and nephronophthisis. Then acquired, that are dialysis associated cystic diseases. Then localized simple renal cyst, renal cyst in hereditary malformation syndrome, including tubular sclerosis. Then glomerulocystic diseases, extra parenchymal renal cyst, including pilocalyceal cysts, hilar, lymphangitic cysts. So these are the cysts, number of cysts that are being enlisted. Now to talk about adult polycystic kidney disease, it's always autosomal dominant and its pathological features include, these are the large multicystic kidneys. Along with these multicystic kidneys, we also find the cyst in the liver, and the patient may present with berry aneurysms. The patient clinically present with hematuria, flank pain, urinary tract infection, renal stones, hypertension, chronic renal failure, and it begins at the age of 40 to 60 years. It's a hereditary disorder that is characterized by multiple expanding cysts of both kidneys that ultimately destroy the renal parenchyma and cause renal failure. As we discussed that it's an autosomal dominant inheritance. Despite that, the manifestation of the disease requires mutation of both alleles of either PKD gene, mutations in genes located on chromosome 16p13 is PKD1 and PKD2 genes. The PKD1 gene encodes a large integral membrane protein that is named as polycystin 1, which has a large extracellular region, multiple transmembrane domains, and a short cytoplasmic tail. Whereas the PKD2 gene, gene products polycystin 2, that is an integral membrane protein. It has been localized to all segments of the renal tubules and is also expressed in many extrarenal tissues. So polycystin 2, it functions as a calcium permeable cation channel, and a basic defect in this PKD disease is a disruption in the regulation of intracellular calcium levels. Dear students, the epithelial cells of the kidney each contain a single nonmotile primary cilium, a 2 to 3 micrometer long hair-like organelle that projects into the tubular lumen from the apical surface of the tubular cells. And the function of that cilium in the tubular cell is it senses the mechanical signals, that is, it's a mechanosensor. Why? To monitor the changes in fluid flow and shear stress. What happens? In response to external signals, these sensors, they regulate ion flux, that is, the cilia can induce calcium flux in cultured kidney of the epithelial cells, and the cellular behavior including cell polarity and proliferation. So the hypothesis that defects in mechanosensing, calcium flux and signal transduction, all these underlie the cyst formation. So my dear students, both polycystin 1 and polycystin 2 are localized to the primary cilium. These polycystin 1 and polycystin 2, they may form a protein complex that acts to regulate the intracellular calcium in response to fluid flow, Perhaps because fluid moving through the kidney tubules causes ciliary bending that opens the calcium channels.